Okay, this video is going to walk you through how we set up our Drosophila cultures, all the way through the napping of the flies and the scoring of the flies. But before you can culture Drosophila, you have to give them someplace to live. And that's going to happen in a vial like this. Ultimately, they don't need any food to eat, but their eggs and larvae do. So that's accomplished by using a potato starch product that comes out of a bag like this. We would take a scoop, like so, put it into the culture vial, then we would hydrate the medium is turning blue because it has a blue dye in it to help us see the eggs, which are white, and the larvae that are white against the contrast of what otherwise would have been just white potato mixture. Now, the entire use of the potato mixture is so that the yeast, which come as little freeze-dried pellets, have something to eat. So the yeast gets sprinkled into the culture. Okay. I like to add a little bit more water to hydrate the yeast. Now, again, life cycle of the fly, which we'll come back to in a little bit, features flies laying eggs. The eggs hatch into larvae, oftentimes called maggots. These are fruit flies, and so everything about them is small, so we're not going to see very large maggots, but we'll see them in a little bit. But that's technically their larval stage. The eggs and the larvae eat the yeast. And in order to raise the yeast, we put all this potato starch in here because that's what yeast like to eat, which is carbohydrates. So in the end of the day, once again, the adult flies don't need to eat anything. They just essentially exist to lay eggs. So we've built this mechanism here so that once again the yeast can breed and consume the potato starch, which then will ultimately give eggs and larvae that are laid by the flies that will go in here something to eat. Now we want the flies to have some place to rest and sit while they're buzzing around inside their culture, so we take a piece of plastic netting like this, and it gets folded in this manner, and then it gets stuffed down inside the culture tube till just above the blue medium, which then will get a plug put on it. And at this point, the culture that will house the flies is fully assembled. Okay, when we come back, I'll be showing you what an actual culture looks like that has the full life cycle of the flies going on in it, and then we will get about the business of transferring living flies from a culture like this to another tube. We'll get them napped, anesthetized, and then scored. So we'll be back. Okay, we're back, and this is just a quick image of the life cycle of Drosophila. This image also exists on your online resource, which you will eventually see if you haven't already seen it. So essentially, male and female flies. Male inseminates a female. Female lays an egg. The egg hatches within a couple days into the larval form, which again, oftentimes referred to as a maggot. They're an off-white little worm-looking guy. And then, much like you may know, caterpillars turn into butterflies. There's a late larval stage and then a pupae stage where the fly is actually forming into what looks like an actual fly inside of what looks like a little cocoon, and then he sort of hatches out of that, molts out of that, and becomes an adult fly. So now I'm going to show you what a culture looks like that's actually mature. Give the camera a second here to focus. And what you'll notice is this looks a lot like what I just made for you before, except the blue is a little grungy because it's been eaten and used for a while. But you see the blue medium, you see the netting, and then you see the flies moving around. And what you see scattered on the sides of the culture tube vial are empty little pupae. And if you can't see from where you are, but when you get your hands on them, what you'll notice is that there are small larval versions of the fly. Here's one here that you may or may not be able to see, and here's one on the side as well. And these are making their way up the side of the vial after having hatched. The eggs are laid on top of the surface of the blue medium. Again, the eggs in the larval form will eat the yeast that are growing in the medium. Then they'll crawl their way up onto the side. They'll make their little cocoon, the pupae stage, and then they'll hatch. And presto, there you have adult flies looking to do the whole thing all over again. Okay, when we come back, we will be transferring these flies to another tube so that we can anesthetize them and then score them, and we'll do a full explanation of that when we get back. And we are back to show you how to transfer and nap the flies. Now, the reason I'm taking the lead with my lab partner, and this is at least a two-person job, is because I'm a freshman who went to a really good private school last year, and we did this lab in 8th grade, so I kind of know how to do this. But I need my lab partner Mackenzie's help to do this. What do you think about this lab, Mackenzie?
It's so great to be a freshman at Redwood because we get to take AP Biology labs as a freshman.
In integrated science.
In integrated science.
I'm happy too. Okay, so here's the concept. These living flies that are in here flying around have to get transferred to this vial where the chemical that's going to put them to sleep is going to be administered. Now, first off, you're always going to want to have a vial that's somehow labeled or identified in your classroom for fly nap. That's what the chemical is called that we use to anesthetize them. So here's a vial that's called fly nap. And then I also have a bag in the classroom that says fly nap on it because these plugs have been exposed to fly nap, as opposed to these plugs and these vials, which are fly nap free. We don't want the anesthetizing chemical to come in contact with the cultures during the course of the 10-day lab because it'll potentially do damage to the larvae and eggs, and it may put the adult flies to sleep, and then they'll fall down and get stuck in the goop. So we have a separate vial for the transfer and a separate cap. And the concept is this. We've got to get the flies away from the cap, so I'm going to tap those down a little bit like this. Don't tap so hard that you drive the flies down into the goop. You just want them away from the cap, because you'll see I'm going to say 1, 2, 3, and on 1, 2, 3 I'm going to pull this cap off, and Mackenzie's going to flip this over the top, and we're going to then transfer the flies, and Mackenzie will take over the next part. So lab partner number 1 takes control of your culture, and they start tapping the flies, and they notice where they are, right? And so then I'm going to do this on 3. 1, 2, 3. Okay, and now I'm going to let go because she has control of that. Now you're going to want to see if the flies should start to fly up into your transfer vial, and there's a few of them right there. And after they've begun to fly, and you have your eyes on who else is still left in there, Mackenzie will then flip the vials over.
So do it now?
Yep, she's going to flip them over.
I think they're all in there.
And now she's going to check the upper vial to make sure there's any flies left in there. She's going to give a little tap for 2 reasons. One, to make sure the ones that are up there are down, and then she also needs to make sure that the transfer flies are low enough in here, because we're going to do this thing in reverse. Now remember, this was the good cap. Here comes the fly nap cap.
There's one in there.
So, and if you can't get them all out, don't tap too hard, because now the goop and the netting may slide into your transfer, and that'd be a total disaster. So she's going to make a decision. Do I have them? Do I have what I can get? She's going to do a 1, 2, 3 also to get the last fly down, and then I'm going to cap it.
All right, ready?
Now, if there is flies up here, you say there is one, right?
There's now 2.
Then I'm going to give her back her cap, because she's going to have to cap that as soon as she pulls it off. I'm responsible for capping the transfer vial. She's going to be responsible for capping this one. Okay, on 3.
1, 2, 3.
Boom. And we have successfully transferred flies. We didn't lose any flies, which is so important. That's not only your data, but these guys are total pests as well.
Yeah, they're in here.
Okay, good. And again, if you can't get them all, it's okay. Actually, once we nap these guys, we can actually do another transfer. So let me show you how we nap these guys, meaning put them to sleep. So there'll be boxes around the room with the chemical called fly nap. And inside the box of fly nap, there'll be what we call a wand. Looks like this. Okay, and then there'll be the bottle of fly nap. And you'll open that up, and you'll soak the wand in the liquid chemical, which gives off a fierce gas. And we're going to do this tapping thing again. Again, I have to open the cap just enough to get this in. So I got to get the flies away from the lid. And I go 1, 2, 3, and I open this up and I stick the wand in and I close it back up. Now, the flies are inhaling the fumes. We getting a little close up on that? Mm-hmm. Good. And it won't take long. They will be affected. They're already not flying very well. Probably in the course of about a minute. The guy at the bottom is already pretty incapacitated. It's a neural blocker. Now they're both still able to climb up the side, but not flying very well. Oh, he just fell off the side. That guy, he just fell down to the bottom. And when they're fully anesthetized, by the way, they'll still be twitching and they're fully alive. They just can't fly. And so we will anesthetize until we're sure that they can't walk or fly. And right now, this guy at the bottom is still crawling around a little bit. Okay? So to give you a sense, if you did leave 3 flies behind like we did and you want to get them, your lab partner can come back, right, if you had to. Yep, so your lab partner can come back, and as soon as you're sure these guys are anesthetized, you can go ahead and take this whole thing apart, and we can go through the whole process again. Right, this time, yep, so I'm going to tap 1, 2, 3. Oh, you know what? My bad. This is really not a good idea. Go ahead and get the flies out of here. It would make no sense to tip these over. Go ahead and get your napped flies onto a scoring card like that. Now we can do it. Okay. 1, 2, 3, go. Boom. All right, go ahead and flip yours over. And give it a little tap, see if we can get those flies down in there. Those guys don't want to come out, huh?
Mm-mm. They're in the
netting.
Yeah. He's on the geese.
Try turning it over. And let them fly up and out. That's oftentimes a trick. They like to escape, so here they come. There we go. Now we're coming up. There's at least 2 of them, right?
There's one more.
Well, we got at least 2 in there.
Yeah.
All right, let's go ahead and yep, turn it over and see if we can get him to come out by tapping. All right. And beautiful. We're going to do this in reverse. Right. This is my fly nap thing, and here's yours. Right. 1, 2, 3. Go. Got it. All right. So you get a chance to see the napping again. Put the wand back in the chemical. Tap my living flies down. 1, 2, 3. Put my finger in. Slip in the wand real quick. Let the gas do its work. You guys can see that my napped flies are sitting right here on my sorting card. My sorting card has an M and an F on it because I'm always going to want to be sexing the flies, determining their gender. And one of the flies is already knocked out. The other 2 are still walking around pretty good, but they're down in the bottom also. Pretty powerful. I'm going to remind you this again in a moment, but keep in mind that a napped fly is still alive. And when he comes out of his anesthetic, he'll fly away, and we don't want any flies released in the room. So, little guy's still walking around down there. The other 2 look like pretty passed out. You're going to want to always keep the fly nap closed as well, so the fumes are not getting in the room as much as possible. And that guy's still walking around, but not for long. Looks like they're all napped. So when we come back, we'll be in the final steps of this video, and we'll be showing you how to score the flies. Okay, so that now that the flies have been napped, and you saw me transfer them once before, I'm going to take these other 3 flies that we took out the second time. I'm going to put my wand away where it belongs in the box with the fly nap. The fly nap is already covered. There's a little spot to put it in here so they can dry out like that. And then I'm going to go ahead and put the cover on, set that aside. Okay. And I'm going to take these flies that we napped and put them back on my scoring card. I have a total of 5 flies there. Then I'm going to make sure that this goes with that, because remember that's a fly nap jar and a fly nap cap. Then I'm going to put the sorting card onto the microscope and take a sorting brush like this, which is really nice for moving the flies around. And you'll learn at another time how to sex the flies, but that all has to be done under the microscope. But there's a fairly simple way of telling males from females. So I'm going to go into the microscope and I'm going to look, and I see that these 2 guys are females, so I'm going to push them over to the F side of my card. I can tell that these 2 are females, and this guy's a male. So now I have them separated at least by male and female, and there'll be other phenotypic traits that you'll be scoring the flies on, meaning taking data. Things like apterous wings, white eyes, red eyes, regular wings, okay? And all that is done also under the microscope. And if you need to move the fly onto its side to see a trait, that's what the little sorting brushes are for. Okay? Now, when the flies are scored, they have to be moved to the morgue, because we're done with them now. We don't want them flying away. The anesthetic will work for about an hour, so we're not really paranoid about these guys waking up. So the morgue lives inside the fly nap box, and it's a vial that looks like this. And you would crack open the morgue, and you would make sure that every fly is accounted for. If you drop one on your pad, just pick it up. The flies get dumped into the morgue. Now I'm not going to actually put these particular flies in the morgue, because this is part of a culture we are still using for the lab. So I'm actually going to transfer them back to where they came from. But if this was the real lab, you get the morgue. There may be only 3 or 4 of them in your classroom in any given period, so you have to ask, like, who's got the morgue? And you go and you get it. It should always remain closed because it has a liquid in it that kills the flies. And then you would pop open the morgue, transfer every fly into the morgue. If you lose one, get it. Keep in mind, a napped fly is alive, and it will wake up and fly away at some point and be a pest in the classroom. So find every single fly. If you counted 5 flies, you should be morguing 5 flies. So the 5 flies get dumped into the morgue. Morgue gets shut nice and tight. Same rules as the the fly nap. Everything's tight, closed, put away, and then these flies would be morgued. And then you could turn off your microscope, and then you'd be done counting flies for that day. Now, this might be interesting to you. I made a point that we don't want the flies in the goop, and these guys are anesthetized. So the technique for putting these guys back is to dump them in gently, and then I'm going to leave the culture on its side like that until they wake up about an hour from now. And then they'll be just normal, flying around like they were before. Okay, when we come back, we're going to show you the rules of the Drosophila Lab for student behavior in the classroom. Okay, here we go. We are back, and we're going to just review the Drosophila Lab rules. Rule number 1, respect the fly. This is a living organism that we are using as an experimental organism in our lab. There's no crushing the fly, releasing the fly, torturing the fly, pulling the wings off the fly, none of that stuff. Respect the fly. Rule number 2, see rule number 1. That's an emphasis about respecting the fly. Rule number 3, no flies escape. That's our data, and they are pests. Rule number 4, see rule number 3, another major emphasis, no flies escape. Number 5, fly nap stays closed. And if you're asking yourself, hey, will the fly nap put me to sleep? The answer is no, but it could give you a headache, and it's not the greatest smelling stuff either. So make sure you cap that fly nap if you're the person responsible for taking the cap off. Number 6, the morgue stays closed. That's going to have alcohol in it that kills the flies instantly as soon as you drop them in the morgue. The morgue's going to fill up with literally hundreds of flies. Keep it closed. True story. Last time I did this lab, fresh sophomore girl, short, tight, white skirt. And the morgue was sitting in front of her with probably about 1,000 flies in it. And it wasn't closed. And the kids are goofing around, and the morgue tipped over the side of the table and right onto her lap. And she freaked out. When she stood up, she had like 1,000 flies stuck to her skirt and a big wet stain from the alcohol. And it was horrifying. So don't let the morgue stay open. Shut it every time you use it. Cool story, yeah? All right, here we go. Number 7, be aware and be engaged. This lab, especially with the living organisms and all the care we have to take about having the flies not escape, you've got to be dialed in when you're doing this lab. You can't be like spacing out and looking around. You got to be engaged in what you're doing, especially when transferring the flies. Number 8, materials as instructed. Very important that you follow every rule in this lab to the letter as it relates to the materials being used. Number 9, all scored flies get into the morgue. Why? Because a napped fly is still alive. You nap 5 flies, you score 5 flies. You score 5 flies, 5 flies go in the morgue. If a fly ends up on the table or on the floor, go get it. If it's napped, later on it's going to wake up and be a pest in the room. Get it into the morgue. And number 10, have fun. This is an awesome lab. Hopefully we'll be collecting data for about 10 days, and hopefully every lab group will get somewhere 125, 150 or more offspring from their crosses. Okay, and that wraps up this video.